Hello, my name is Marie Cruz and I'm going to be talking about systemic lupus erythematosus, or SLE for short. So what is lupus? Lupus is an autoimmune disease associated with the presence of autoantibodies targeted against nucleoprotein antigens. So as we know, an autoimmune disease is when the body's immune system starts attacking itself instead of only attacking foreign invaders. So who can get lupus? Anyone can get lupus, but it mostly affects females of African American, Hispanic, Asian, and Native American descent. So females may get the disease more because of estrogen, which we know can cause different effects on the immune system. So there are different forms of lupus. The most common form is systemic lupus erythematosus, or SLE for short, but the other forms are discoid lupus erythematosus, subacute cutaneous lupus erythematosus, drug-induced lupus, and neonatal lupus. I will be just focusing on systemic lupus erythematosus. So the causes of lupus are unknown. The direct causes of lupus are unknown, but research has indicated that one's genes can affect how lupus is triggered, but genes alone do not cause this autoimmune disease. Lupus is likely triggered by many factors. So these are the common symptoms of lupus. There is fever, photosensitivity, ulcers in your mouth and nose, aches in your muscles, arthritis, fatigue, loss of appetite, a butterfly rash on your face, inflammation, and poor circulation to your fingers and toes. How is lupus diagnosed? So someone can get diagnosed of lupus by medical history, a complete exam, a blood test, skin biopsy, or kidney biopsy. As for treatment, there is no cure, but treatments can include taking drugs to reduce swelling or prevent or reduce flares, help the immune system, or reduce or prevent damage to joints or your organs, and it can also there can also be treatment to balance your hormones. So what are flares? Flares are periods of time when the symptoms show up and increase. Symptoms of a flare usually is feeling more tired, a rash, a fever, having a stomachache, headache, or dizziness. And from the previous slide, this is an example of the butterfly rash. So what generally happens in the immune system? As we know, many things can happen with the body and the immune system to activate SLE. In this example, this is just a general overview of how lupus works, and it may be slightly inaccurate, but in this example, complement is shown as this C5 in this picture, and it sends danger signals to this antigen-presenting cell. This antigen-presenting cell, along with these other co-stimulatory factors, then activate this T cell, which sends cytokine signals to activate this B cell, which then will release autoreactive antibodies, which will cause the immune system to go haywire. So this other antigen-presenting cell also stimulates this B cell, causing it to have more proliferation and more differentiation, and it'll produce more autoreactive antibodies, which will then cause an increase of disease symptoms. This all will be discussed later. As I mentioned earlier, the direct cause of SLE is still unknown, but we do know that there are many factors involved with the autoimmunity of lupus. One of those factors researched is a deficiency in complement. So this MHC class 3 region is a region in the genome that encodes for some molecules that are important for inflammation. The MHC class 3 region also encodes for C2 and C4, and they are part of complement. A deficiency in C2 and C4 can also explain or contribute to the risk in those of ethnic backgrounds. So a decrease in complement activity could promote disease susceptibility by impairing the neutralization and clearance of self and foreign antigens. This means that our body can generally have a hard time clearing autoimmune cells, as well as our foreign cells that don't belong in our body. A deficiency in more specifically C1, C4, and C2 is the strongest genetic risk factor for the development of SLE, And even more specifically, a deficiency in C1q, C1R, and C1s gives us an even higher risk of developing disease. So through research, it is found that having a deficiency in C2 actually gives us less of susceptibility compared to a deficiency in C1. So another possible cause of lupus is estrogen. An abnormally high amount of estrogen can cause a higher Th2 response, and a lower Th1 response can also cause autoimmune B cells to be produced, along with the autoimmune antibodies being produced from those autoimmune B cells. Complement can be involved with clearing apoptotic cells. So if we have a deficiency in complement, this can prevent the clearance of autoimmune B cells, which then can lead to autoimmune antibodies being produced from those defective B cells. So part of the research that I found suggested that the early part of the classical pathway can play a part against SLE. So the complement pathway is mainly initiated by the interaction of C1q and the antibodies IgG and IgM, which are the main effectors of antibody-mediated immunity. A C1q deficiency is the strongest known susceptibility factor of SLE. Information suggests that the early part of the classical pathway plays a role in protection against the disease. There is a hypothesis that a deficiency with the classical pathway predisposes us to SLE because it impairs our resistance to infectious trigger, though there is little evidence for this. A deficiency in the classical pathway can predispose us to more bacterial infections, but the bacterial infections are not the cause of SLE. So this study was done to show that the heads of C1q will bind to apoptotic cells. The test was done with the head and the tail, and the anti-tail fab had no effect on C1q, while the anti-head fab did have an effect on C1q, and you can see that there is much less C1q in this figure, and this research did help confirm that C1q did bind to apoptotic blebs via the head. So patients with active or severe SLE have shown to have lower concentrations of these complement components, C1q, C2, C3, and C4, and it has been reported to correlate with the disease activity. So the less complement there is, the more disease activity someone will have. Knowing this, it can also be possible to predict the disease flare-ups in a person. So this picture just generally shows where the C1q is located when they bind to an apoptotic cell. They are indicated by these lighter colored regions of these pictures. The more lighter the colors are, the more C1q is bound to this apoptotic cell. So we'll see that the C1q is bound to this apoptotic HUVEC cell, or the human umbilical vein endothelial cell. But as you can probably notice, they only bound to the surface blebs, or these bulges of the apoptotic cell. These surface blebs on the apoptotic cell is one factor that can cause the immune system to respond. So autoantigens can accumulate here, which can cause the activation of dendritic cells or monocytes, or just antigen presenting cells in general, and B cells, which can release autoantibodies. So it makes sense how a deficiency in C1q can lead to problems with an overactive immune system. As we saw earlier, there are some cytokines involved with SLE. The ones we see here are IL-2, IL-6, IL-10, IL-12, and there are some others like interferon gamma. We know that IL-12 is part of the Th1 response, but the production of IL-12 in a predominantly Th2 response can also indicate that there is an abnormal Th1 response going on. Other studies suggest that there is actually an increase of IL-10, which suggests a stronger Th2 response instead, and since IL-10 deregulates the Th1 response, this can suggest that IL-10 is causing a defective Th1 response, which causes an immune response in our body. So evidence leads closer to an overproduction of IL-10 rather than IL-12 as I began to look at data from more modern studies. So knowing this, an excess of IL-10 production can cause an impaired production of IL-12. And basically, ultimately, I found that in a good amount of studies is that there's evidence for both type of reactions, and concluded that SLE is caused just by an imbalance of cytokines. The Th2 cytokines mentioned, as we know, are responsible for B cell mediated immunity. So in general, there is known to be an IgE production, and as we learned with allergies, we know how this will affect the body. There's also an activation of eosinophils and a deactivation of monocytes. So this picture to the right just shows us many different cytokines that were tested and if they were related to lupus. The cytokines IL-12, IL-17, IL-18, and IL-4 are elevated in the patients compared to the controls, which lead us to believe that these cytokines are involved. Below that is the result of another experiment. This data shows us the ratios of these cytokines. So the ratios of these cytokines compared to the disease activity. So SLEDAI just stands for Systemic Erythematosus Disease Activity Index, which is just the disease activity. So it's just an experiment to see if the cytokines could be related to the cytokine ratios could be related to the disease activity, as we see with IL-18 and the ratio of IL-4. This has some correlation with IL-17 and IL-4 related to disease activity. There is very little correlation and closer to no correlation. IL-12 and IL-4 related to disease activity. There is some correlation, but still kind of like no correlation. So we know that Th1 and Th2 cells usually deregulate each other in order to maintain a balance of the body. An abundance of many different cytokines in the body will not be a good thing for our bodies, and it is suspected that the imbalance of the T helper cytokines can be responsible for polyclonal B cell activation. So some things to mention: IL-18 induces interferon gamma, the production in IL-12 activated T cells and NK cells. This can upregulate Th1 cytokines, so it's just basically the reaction of what's happening. Reiterating the fact that there are many different factors that can stimulate lupus, this is another test that explores the idea that more dendritic cells, or antigen-presenting cells in general, creates a response in our immune system, ultimately leading us to lupus. So what they found is IL-10 suppresses the differentiation of monocyte-derived dendritic cells. So basically, if you have an elevation in IL-10, you can prevent monocyte-derived dendritic cells, and this can contribute to a wrong response that our body has. So this is just a test that uses different types of serums to see how much dendritic cells are being made. So stimulation index is just the degree of proliferation of a monocyte derived into a dendritic cell, and they are testing with different types of serums. So these dendritic cells were induced by normal serum, then they were induced by SLE serum, then they were induced with IL-10 neutralized group, which is just SLE plus IL-10 plus IL-10 neutralizing antibodies. The dendritic cells were also induced by this isotype group, which is made of SLE with rapid anti-human IgG controls. This stimulation of dendritic cells by normal SLE serum with non-neutralized exogenous IL-10 was still present, but not as much as the others, assuming that the higher levels of IL-10 can regulate the amount of monocyte dendritic cells being produced, and we will see later how this will be a problem. So according to research, SLE can, as we saw, can induce monocyte differentiation from a monocyte into a dendritic cell. So this also secretes more cytokines, and we know them as BAFF and APRIL. So this is just a test experimenting on that. So mostly monocyte and dendritic cells. We know this isn't a good thing if we have an overabundance of these, but yeah. This data is based off of an ELISA test that researchers conducted. This figure shows the amount of APRIL and BAFF secreted by monocytes, interferon-induced dendritic cells, as well as SLE-induced dendritic cells. As we see, these induced dendritic cells do produce BAFF and they do produce APRIL. Now that we know that BAFF and APRIL are secreted by monocytes and dendritic cells, we will see that an abundance of them can be bad, as they will cause a failure to maintain B cell tolerance. So this can mean that B cell development is defective at several points, like B cell development, maturation, and the activation of the B cell. So BAFF stands for B cell activating factor, and APRIL stands for a proliferation inducing ligand. So these are the receptors that they are bind to, and the picture on the right is just how they bind to those receptors and where they are located on the B cell. So this is an experiment of BAFF and APRIL, and we can see that there's elevated levels of BAFF and APRIL compared to the healthy control. So along with a deficiency in complement proteins that causes other symptoms, high levels of these cytokines can cause proliferation of B cells and can stimulate more autoantibodies. So this correlates to the CD19 B cells, and the B cells in those with SLE were elevated along with the production of autoantibodies. And then eventually you have a positive loop of B cells. So interestingly enough, one of the treatments of SLE is inhibition of BAFF with monoclonal antibodies. So keep that in mind. In conclusion, there is no solid answer to what causes SLE. We know that something went wrong with our genes that caused the complement system to go haywire, and also causes our B cells to go haywire, and this can have many effects. B cells can cause autoimmunity, which then causes autoantibodies, and just in general, many different factors are involved. So, and more research has to be done on many aspects of the disease to understand what is truly going on. And that is my presentation on SLE, and these are my references. Thank you.